Hello friends, welcome to the student world. Today we will discuss about ligation through homopolymer tailing. Now, what is homopolymer tailing? Homopolymer tailing is an approach by which two non-compatible ends can be joined to each other. For example, a blunt-ended DNA molecule can be ligated to a DNA molecule which have sticky ends. By homopolymer tailing, we introduce sticky ends to a blunt-ended DNA molecule. Now, what is homopolymer? Homopolymer is a polymer in which all the subunits are same. For example, a DNA strand which is made up of only one nucleotide. For example, deoxycytosine is a homopolymer of deoxycytosine, which is written as polydeoxycytosine or poly-DC. Tailing is the repeated addition of nucleotides to the 3' end of a double-stranded DNA molecule. Now you see in the diagram the synthesis of a homopolymer tailing. There is a double-stranded DNA molecule and we have to add poly-DC tail at 3' end. We provide DCTP and the enzyme terminal deoxynucleotidal transferase. This enzyme add repeatedly CCC nucleotide at 3' end and generate poly-DC tail. Now we know about the enzyme which catalyze homopolymer tailing. Homopolymer tailing is catalyzed by the enzyme terminal deoxynucleotidal transferase. It is a template independent DNA polymerase. Template independent means this DNA polymerase doesn't require a template for the synthesis of DNA. This enzyme catalyzes the addition of deoxynucleotides to the 3' end of a DNA molecule. This enzyme prefers to use DNA molecule with 3' protruding end as acceptor. Although this enzyme can also add tail to the recessive ends or blunt-ended DNA, but it prefers 3' ends which are extend out. Now, how we generate 3' protruding end for addition of homopolymer tail? There are two ways by which we can generate 3' protruding end on a DNA molecule. First, by digestion of DNA molecule with a restriction enzyme which produce 3' protruding end. For example, PST1 restriction enzyme. This restriction enzyme produce 3' protruding end after cleavage. Second, by giving lambda hexanuclease treatment. This enzyme is a hexanuclease which catalyzes the stepwise removal of nucleotides from 5' end of a double-stranded DNA. When 5' end is trimmed, 3' end is automatically extend out. Now, we ligate a blunt-ended insert DNA in a vector which have sticky ends. Now step first, in ligation, first of all, the insert DNA with blunt-ended DNA is given lambda hexanuclease treatment so that 3' protruding ends can be generated on this insert DNA. In second step, we add poly-DC tail at 3' ends of this insert DNA with the help of enzyme terminal deoxynucleotidal transferase. You see in the diagram, poly-DC tail is added at 3' end of this DNA molecule. Now, in step 3, we have to ligate this insert DNA with poly-DC tail in a vector. Now, as we know that there is complementary base pairing, so there is base pairing between C and G. We have to add poly-DG tail to the vector. For tail addition, first of all, we have to generate 3' protruding end. Now you see in the diagram there is a vector with PST1 restriction site. When we digest this vector with PST1 restriction enzyme, 3' protruding ends are generated. After generation of 3' protruding end, we add poly-DG tail to these ends with the help of enzyme terminal transferase. Finally, we have to ligate this insert DNA with poly-DC tail in a vector with poly-DG tail. First of all, there is complementary pairing between G and C, and the remaining nucleotides are filled by DNA polymerase. This is known as gap filling. After gap filling, DNA ligase seal the nick. In this form, recombinant vector is formed. Now, what are the advantages of ligation through homopolymer tailing? You see, during homopolymer tailing, each DNA molecule carries the same type of homopolymer tail. For example, in insert DNA, we have added poly-DC tail. Now each molecule of insert DNA will carry poly-DC tail. Same type of tail cannot hybridize with each other. Thus, after cloning, each transformed cell have desired recombinant DNA. There is no hybrid of insert DNA or vector DNA. This is the advantage of ligation through homopolymer tailing. That is all about ligation through homopolymer tailing. Thank you. If you like the video, please subscribe.